Hi, and welcome to this video where we're going to be going through all the key features of biological membranes, which is OCR Unit 2.5 from the OCR A-Level Biology syllabus. Okay, so the first thing that you need is you're going to need the resources that I've sent you on Microsoft Teams. I just want to show you the two different versions. If you've got access to an A4 printer, then print this document off. And then you can cut out all the different pictures and then you can use them on an A3 sheet of paper. So in your exercise book, for example, if you open up a double page to make your mind map. You're also going to need colourful pens and pencils and markers or whatever to try and make your mind maps as snazzy and colourful and memorable. If you have access to a A3 printer, then fantastic. And there's this version as well, which you could label. So the finished version is going to kind of look a bit like this, but we're going to have a lot more information in it. So if you need to pause the video, do so now. Go get all your materials ready, and then come back to me when you're ready, okay? Let's get started. All right, so first of all, we're going to look at this biological membrane over here. So this is the cell surface membrane. Let's get it a nice big title. Wow, that's a bit too big. Cell surface membrane. And it's important that we refer to it as the cell surface membrane, not just the cell membrane, because there's loads of membranes in the cell. You have the endoplasmic reticulum, smooth and rough. You have the membranes around the mitochondria. You have the nuclear envelope. There's a lot of other membranes in the cell, and we're talking about the surface one now. So that's the cell surface membrane. Now we say that it is a fluid mosaic model. And that's because the main component of the membrane is a phospholipid. So this is a phospholipid down here. But there's other stuff stuck into it, like a mosaic. A mosaic is a kind of artwork created by sticking lots of little tiles together in a pattern. And that's kind of what a membrane's like. It's got lots of phospholipids stuck together, held together by hydrophobic and hydrophilic interactions, and then there's other things embedded in that membrane. So the phospholipid, the main constituent of the membrane, we looked at when we looked at biological molecules, but quick review, it has two main areas. It has the polar head group. And the polar head group, key thing, it is hydrophilic. And then it has the non-polar tails, non-polar fatty acid tails, and those bits are hydrophobic. I'm just going to label that here because I'm out of space. Hydrophobic. So you can see that how the phospholipid molecules arrange themselves is they arrange themselves into a bilayer. So we call this the phospholipid bilayer. And if we were to look into that bilayer here, in the middle we've got the hydrophobic tails sandwiched together. So there's no, I'll just put no H2O here. I don't know if you can see that. No, let me do that again. So there's no water in there. So we've got a hydrophobic environment where the tails can kind of get together. Let's label that here. No H2O in the center. Hydrophobic, tails crammed together. But we've got an aqueous environment both inside and outside. So I'll just maybe label this up here. Aqueous. Aqueous environment both outside and inside the cell. So this is the outside. And this is the inside. And I know that because of two reasons. I know because these groups are pointing out. We'll talk about them later. And also I can see these strands, these kind of filaments inside. We'll talk about those now. We'll just label those. Those are called the cytoskeleton filaments. I'm going to label that in blue. Cytoskeleton filaments. And these tend to be on the inside of the cell. And they kind of give the membrane the shape. They're responsible for the shape of the membrane. Without them, the cell, I guess, without them, the cell would be kind of probably a perfect sphere. But sometimes the filaments can distort the membrane so that the cell might be kind of this shape, or gives it a certain specific shape. So those are the cytoskeleton filaments lying just underneath the cell surface membrane. Okay, what else shall we look at next? All right, well, let's look at the main things that are embedded in the membrane, the different structures. We've got proteins, okay? Lots and lots of different proteins. I'm going to label the proteins in black. So this could be a carrier protein here. Embedded into the membrane. These proteins here are channel proteins, channel or carrier proteins. And these are called integral proteins. And that's because they go all the way across the membrane. They're stuck in there, they're embedded inside the membrane. So they have membrane spanning regions, whereas this one is a peripheral. Oh gosh, am I going to be able to fit this in? Peripheral protein. And that's because it's not actually in the membrane, it's associated with a separate protein. This protein would be integral, right, int. But this one is peripheral, because it's associated with the membrane, it's on the outside of the membrane. Okay, so we're going to look at what these channel proteins and carrier proteins can do and the difference between them in a minute. So I'll just kind of link these to them. So this is a carrier protein. We're going to look at it here, carrier. And this one is a channel protein, which we're going to look at here. Oops, that's the bilayer there. So this is channel. Okay, so we'll look at those a little bit more detail later. But briefly, they allow molecules into and outside of the membrane. Over here, this one in your textbook is labeled a little bit more detail. So this is a channel because it has a water-filled channel or pore that links the external cellular environment to the internal cellular environment. Okay, so this is an open channel. And channel proteins, actually, they're not sort of filled with water. They can actually be very specific. So channel proteins can select which ions they allow down the center. So potentially, I'm going to zoom in a little bit more. This is going a little bit beyond that level of detail that we typically do at AS. This is kind of going into university level, but around this entrance point here, there might, for example, be certain R groups on amino acids that stick out. So maybe these R groups are negatively charged, potentially, and maybe they're a certain distance away. And that would mean that only a positively charged ion would be able to go down there. And it can actually even discriminate between different positively charged ions, so maybe between sodium and potassium, for example. And there would also be other channel proteins that might have positively charged R groups here, which might only allow negative ions through. So the channels can really select which ions or molecules can pass through them, and that's because of their 3D structure, essentially. Okay, all right, so those are the proteins, but there's some stuff on the outside of the membrane too. So what on earth is that? Well, these are basically carbohydrate groups. Here's another protein. What does it say? Could be an enzyme. We can't really tell actually from this image whether it spans the membrane or not, because it's kind of hidden behind the surface that we're viewing. So I'm just going to label that as a protein. Perhaps it's an enzyme. Here's a protein as a receptor site. So maybe let's label onto that. You know, maybe a hormone might bind there. Hormone binding. We're going to learn a lot about that in year 13. So maybe that's what happens. A hormone binds that receptor. Perhaps it's an insulin receptor, maybe. Okay, now what about these yellow hexagons? If you think hexagons in biology, you should be thinking generally carbohydrates, because carbohydrates are generally made of the hexosugar glucose. So these are all carbohydrate groups, but there's slight differences to what they are. So this, let's label all the glyco bits with green for me. This is a glycoprotein. Now, why is that? Well, it's because these carbohydrate groups are attached to this protein down here. Can you see that one there, this purple shape down here? That is the protein bit. This is the glyco bit. So together it's a glycoprotein. This one here, it looks like this is attached to a lipid. That's attached to that one right there. So that's a glycolipid. And in your book here, this looks to me like it's a glycolipid again because it's attached to a lipid. But in your textbook here, this is just a bit more generally about the word glyco. So glyco does mean carbohydrate. So it's carbohydrate groups. So these carbohydrate groups kind of project outwards from the membrane. And there's another really weird word which you should be aware of. And it's that these glyco groups, these carbohydrate groups that project outwards, form something called the glycocalyx. Okay, so I'm going to where am I going to put that? I'm going to kind of put this up here, like this, like that, and this is going to go over here. So this is the glycocalyx. Now, don't use this word that much, but it could come up. So I'm just going to put that like that, glycocalyx. So this is basically the carb layer. And it sits outside the membrane. It's kind of all mainly to do with cell-to-cell recognition. So it helps the cell interact with its environment, and it's involved in recognition, for example, cell-to-cell recognition and interaction. So cell-to-cell recognition, interaction with environment, etc. Okay, so that's the cell surface membrane. Now, one thing you should be just a little bit aware of, again, a little bit of extension, is how do we know it's fluid? How do we know things are moving around? I'm going to describe for you one of the experiments that was done. You don't need to know this in detail, but it's something that you might be given the information about, and then you have to sort of draw conclusions from it. It's actually called the fluorescence recovery after photo-bleaching experiment, which was pretty interesting, actually. So basically what they did was, first of all, they stained all the phospholipid molecules with a fluorescent dye, okay? Let's say they stained them blue. Look, here they are. They're all stained blue. All the heads are stained blue, like so, okay? They all stained them blue. Now what they'd use was they use a very, very precise laser to target those phospholipids and bleach the blue dye which they'd attached. And they bleached it in a very certain spot. So they did this, basically. Okay, let me just see if I can do this quickly. So they bleached, yeah, it's going to work. Look, they bleached all these phospholipids over here with a laser. Okay, the laser bleached them. There they are. Ta-da! Okay, in that little pulse. They then waited like a second, okay? And then they went back and they took another image of the cell to see those phospholipids that they bleached, are they still there? And they weren't. So what happened then, let's get rid of those, is when they looked a second later, they found that those phospholipids had sort of spread. They'd sort of diffused laterally, and they'd moved about a bit, okay? And then maybe they came back a few seconds later and the sort of phospholipids had spread even further. So we know that the phospholipids are constantly jostling past each other. They're moving, they're fluid. So all these proteins can kind of diffuse along left and right. They can't pop out of the membrane. They're held in that two-dimensional plane, but they move about. So that's why we know that the that we call this a fluid mosaic model. Molecule here, which I also noticed that I haven't labeled, and I've sort of run out of space, so I'm going to label it down here, is cholesterol. Oh, whoops, that's way too big. Let's make that smaller. There we go. Is cholesterol. We talked about that in the last lesson, but remember we said that it regulates membrane fluidity. Okay, so that is an overview of the fluid mosaic model of the cell surface membrane, okay? So if you'd like to now, you can have a look at that in your textbook, page 128, 129, read through it. You can add some more detail if you feel like you need to. And next thing we're going to do is we're going to move on and we're going to look at how do things get across membranes. Okay. The first thing that you need to understand about how things get across membranes is that not a lot can without help. So the membrane is an effective barrier, so it can effectively compartmentalize the inside of the cell from the outside of the cell. That's the whole point of the membrane, really, is so that there can be different concentrations of molecules inside the cell as there are on the outside of the cell. But there are times we need to move things across. So first of all, let's think about diffusion. Okay, diffusion, simple diffusion. That is the easiest way, or the simplest way, that things can get across the membrane. So diffusion. So diffusion happens from a high concentration to a low concentration. So this is just diffusion through the phospholipid bilayer. So what kind of things can go through the phospholipid bilayer? Let's just label this up here. Let's show a molecule diffusing. I'm going to make let's do a black blob. Let's say a molecule over here. Where's space? I got no space. Let's do it here, okay? Blob. Okay, this black blob here, it can just diffuse straight through there into the inside, okay? And then here's the same black blob. So it moves from a high concentration, so let's do a few more black blobs up here, high concentration to a low concentration, down its concentration gradient. But what type of molecules can do that? Well, there's a couple different molecules that can diffuse. So if it is a small, non-charged molecule, okay? So, for example, water can do that. Oxygen gas, O2, CO2 gas. Those are all small molecules, but they're not charged. They might be polar, but they're not charged, okay? So small non-charged molecules can. If it's a charged molecule like an ion, it's not going to do it, okay? What else? Also, large, I'm going to run out of space there, so I'm going to do it down here. Also, large lipid-soluble molecules. Can diffuse through the phospholipid bilayer. So, for example, a hormone, okay, hormone testosterone, that is a lipid or estrogen. Those are both lipid soluble, and they can actually just diffuse all the way through the center of the bilayer because they can dissolve in fat, and the bilayer is essentially fat, okay? So small non-charged molecules, yes, they can go through. I'm going to put a little tick, tick, tick. Small non-charged molecules can do. Tick. Large lipid-soluble molecules can go through, and let's put a little cross here. Cross. Ions. Ions can't go through because they've got a charge. Or cross large moles, e.g. glucose, okay? Glucose can't diffuse through. It has to get through a different way. So, that's diffusion. So diffusion, I've kind of slightly gone over this little diagram here. This shows that we've got out here, we've got high concentration. This is the membrane. And this is a low concentration, okay? So diffusion happens from high to low across a membrane. This graph here kind of represents the gradient. So this is the high concentration here, and this is the low concentration here. So moving from a high concentration to a low concentration across a membrane. That is what diffusion is. Now diffusion depends on a few things. It depends on all these things listed here. And they're outlined in your book, but I'll run through them quickly. So as the temperature goes up, it means molecules moving more quickly and diffusing more quickly. There's also a slightly different effect, which we'll look at in more detail later, which means that if it gets too hot, the membrane becomes more permeable because those phospholipids are moving about so quickly that they start to have little gaps in them for just a little microsecond where a particle can get through. So temperature increases the rate of diffusion. Now, diffusion distance, this decreases the rate of diffusion, okay? Because if something's got to move over a longer distance, it's going to take longer to do it. Surface area, this is going to increase it. Higher surface area equals faster diffusion. And we find cells that are adapted to have a specifically high surface area. For example, intestinal epithelial cells have sort of brush border like this, microvilli, so that they have a high surface area for diffusion. And also other transport processes, okay, high surface area. The size of the diffusing molecule, the smaller diffusing molecules diffuse more quickly. Oops. And concentration gradient. Higher gradient equals faster. Okay, so diffusion depends on all of those things. Temperature, diffusion distance, the surface area, the size of the diffusing molecule, and the concentration gradient. But remember that diffusion across the membrane is only a way in if the molecule is small and non-charged, like small gases, if it's larger and lipid soluble, but if it's a small charged molecule like an ion, it won't go through, or if it's a larger molecule like glucose, glucose isn't charged, but it's large enough that it won't really be able to pass through the membrane. So what about those molecules? Sometimes we've got to take glucose into a cell. So how does that happen? Well, at this point, we've got to use something not diffusion, but facilitated diffusion. So I'm going to move up to this part of the Mindmap here. And we're going to label, let's go a little bit bigger. Is that about right? Yeah, that's about right. Facilitated diffusion. Okay, so facilitated diffusion can involve either a carrier or a channel protein, okay? And we say that it's facilitated because it's helped along. So facilitated, that word kind of means helped along. So it's diffusion that has to be enabled by a protein. So let's call it enabled, okay? Enabled by a protein. So here is a protein. This is a carrier protein, and this is a channel protein. Now, we've already sort of talked about the channel protein already. We talked about it here in a little bit of detail. So this might be an ion that is allowed to go through, and I'll just write here, it can select which ions it allows through due to the specific 3D structure. Other things to label here, we've got the phospholipids, phospholipid bilayer. This is inside, this is out. And actually in this little diagram here, we can see this is a, these are some molecules that are diffusing just without the channel. So this would be, let's say this is a small molecule like O2, for example. And maybe this is an ion that goes through a channel. Ion channels are very common types of protein in the surface of a cell. Okay, so that's channel proteins. Let's highlight that actually, this channel protein here, the difference. Okay, channel protein. Now, what is the difference between a channel protein and a carrier protein? The main difference between a carrier and a channel protein is that the carrier protein changes shape somehow during the transport. okay? We sometimes say that it shows a flip-flopping, okay? So a flip-flopping is like this, okay? It's facing one way, then it flips, and it's facing the other way. One way like this, like that, okay? That's flip-flopping. So let's label that flip-flopping. Okay, let's zoom in a little bit so I can explain how this carrier protein works. So let's say that this carrier protein is taking in glucose molecules. So let's say this is glucose. All these molecules here are glucose. Right, so the glucose, first of all, the carrier protein is kind of in its conformation where it's flipped and it's facing out, okay? So this opening is facing out of the cell. So here's the opening and the glucose goes in and binds. Right, I'm gonna have to try and write really, really small here. See if I can do it. Glucose binds to protein. When it binds, it triggers what's called a conformational change. So it changes shape. The binding causes the protein to flop like that. And now that glucose is taken through and it's released inside. Okay, so this from here to here is kind of happening in time. This is just at the beginning. This is tiny microsecond later. So this would be labeled a conformational change. Conformational, spelling mistake. Conformational change, not conformational. Conformational change. Basically, it changes shape, changes its 3D shape. And now that glucose molecule is released. Okay, so now that is now released, glucose released. Okay, once that glucose molecule is released into the cell, the protein will kind of flip back again. So it flips, glucose binds, flops. Glucose releases, flips, glucose binds, flops. So flip-flopping like that, okay? That is the carrier protein sort of mechanism. Other thing to label in this right here is, of course, the phospholipid bilayer. Again, this is, again, the phospholipid. This is a bit of an odd diagram. One thing I don't like about it is there's a big gap here between the phospholipid tails. That I don't know why that is like that. It shouldn't really be like that. You just join those up if you'd like. It's strange. Let's just extend those out. Okay, it should be like this the whole way. Okay, there's no gap in the middle. I'm just gonna quickly do that. Better. Okay, so that's facilitated diffusion. It's important to note, I almost forgot, that facilitated diffusion is a passive process. Let's do that in blue. It's still diffusion. It is a passive process, which means no energy, or brackets ATP, because that's what energy is in the cell, is needed. Okay? So this flip-flopping, you might say, oh, so there's got to be some energy because these proteins are changing shape, they're moving. The energy really to make the proteins flip and flop, if you want to be really technical about it, the energy comes from the concentration gradient. So it comes from the molecules moving down their concentration gradient, kind of causes the flip-flopping. Okay, so there's no extra energy required for this facilitated diffusion. If something needs energy, we have to use active transport. Okay, so why would it need energy? I need to add one more thing here. So facilitated diffusion, passive process, no energy is needed, and it is always down a concentration gradient. Okay, that's why it's diffusion. Active transport can move things up against the concentration gradient. So let's travel down here and look at a type of active transport. So again, let's label this one slightly large. And yeah, let's do it like this. Active transport. Okay, so active transport, let's contrast it with what we just looked at. So active transport, up a concentration gradient, That means we're moving something from low to high concentration. And what else? It requires energy. And that is in the form generally of ATP. ATP, we'll learn about it more in year 13. It's called the kind of energy currency of the cell. It's how the cell kind of pays for processes that require energy. So ATP is needed. And one of the proteins that uses ATP to transport things is the sodium-potassium pump. This is a really, really important protein in the cell. So some estimates have said that the action of this pump actually uses around about 55% of the ATP that a cell uses. Now, I had a little look into that. I'm not sure if that's specifically for some type of cells. It certainly is true, I think, of neuronal cells, because a lot of the energy they use is used to kind of transmit electrical impulses, and the active transport by sodium potassium pump is used to kind of set up the electrical charge that can then be used to trigger to send those impulses. So up to 55% of a cell energy use might be just from this pump. Now, this pump is so important that I'm just going to stop talking for a minute and let you just look and see how it works. Okay, so just watch this video. It will repeat round about 12 seconds. Watch this video, and then I'm going to take it away and we're going to talk about the sodium potassium pump. So see if you can identify what ions are going in, what ions are going out, and at what point ATP binds. Okay, let's take that away, and now let's label the different stages of that process. Okay, so the stages of that process. First of all, this first box here, I'm going to draw a little line down here, is that we have 3 times sodium ions bind, and so does ATP. And we have the hydrolysis of ATP into ADP changes the shape of that protein. So we have a conformational change of shape, and then the protein changes shape. I'm just going to change color to make it kind of clear which stage is which. So ATP converts to ADP. We call that hydrolysis of ADP. And we have a conformational change of the shape of the protein. So now that the protein has changed shape, it's kind of flipped, and now the sodium ions can be released. So I'll change color again. I can make it a bit smaller actually. So sodium ions released externally to the external face of the membrane. And then the final stage is that it's the binding of 2 potassium ions triggers a conformational change in shape back to the starting shape again. So it kind of constantly cycles through. Oops. So I suppose the key point of this whole thing is this stage here. It's the ATP hydrolysis to ATP. That's the key thing. That's why it's active transport. I think it's important to note, just in case you were thinking, are sodium ions and potassium ions circles and triangles? That's just for diagrammatic purposes. They're just circles. I mean, they're basically spherical ions. They're slightly different sizes. So that's how the kind of protein can kind of tell them apart if they're both positively charged little blobs. Okay, so that's active transport and a membrane, but there are also other forms of active transport that kind of work on a larger scale. So let's look at that. So let's take a line from this active transport over here, bigger line. And let's also talk about these two forms of active transport as well. So these two forms of active transport, sometimes you can call them bulk transport. Okay, so bulk transport means B-U-L-K, bulk transport means you're moving larger quantities of stuff. You're not just moving ions across a membrane to or fro, you're moving loads of stuff. So two key processes you need to know about: exocytosis and endocytosis. So exocytosis moves things out of a cell, exocytosis, and endocytosis takes things into a cell, takes things in bulk quantities into a cell. So let's go into a little bit more depth about exocytosis and endocytosis. So here, this might be a food particle. Oops, I'm going to make that a lot smaller. So that could be a food, or it could be something like a bacterium. So white blood cells use a modified version of this endocytosis to kind of to engulf bacteria, and then they digest them. So it's kind of a modified version of endocytosis. So food or bacterium, the cell kind of moves its membrane around the outside of the thing it wants to swallow, and then it takes it inside, so to form an endocytotic vesicle. Label that here, endocytotic. What's an adjective? Yeah, endocytotic vesicle. Basically, it means a little bubble of stuff that the cell has just kind of swallowed, if you like. Exocytosis is kind of the reverse. It's a little bubble of stuff in the cell that then goes towards the membrane, fuses with the membrane, and releases into the external environment. So that's what we see here. Number one is the vesicle. Could call that the exocytotic vesicle or transport vesicle. Here, stage 2, we've got it fusing with the membrane, and 3, we've got release of the products. Now, because these are both active transport processes, they do require ATP. We won't go into the details of how ATP is used, but the reason they're distinct from the sodium potassium pump is because they're happening on a much larger scale, bulk transport. Okay, so those are the key ways that things get across membranes. We talked about passive processes, we talked about active processes, but there's a key passive process that we haven't talked about yet. We haven't done so because it gets a little bit more complicated. So another passive process, the way that things can get across membranes, is of course osmosis. So let's do another big title, osmosis. Right, so osmosis is all about movement of water. So let's make a little, so actually let's write the definition, okay? It's worth it. So the definition for osmosis, the definition for osmosis at A level is a bit more complicated. It's the movement of water from an area of high water potential. This is slightly different than GCSE. An area of high water potential to an area of low water potential across a partially permeable membrane. Okay, so that definition is pretty complicated. Let's highlight the key areas. So first of all, we need to use the word water potential. and not anything else. So let's highlight that, water potential, water potential. Second of all, let's highlight the fact that we're talking about high to low. So that tells us that it's a passive process, high to low. And the third thing, let's not forget that we're talking about across a membrane. It's a partially permeable membrane. The reason we say it's partially permeable because it's permeable to water and other things, but not everything. So let's analyze that statement through looking at this picture right here. So what is this picture right here? So, okay, so here's our membrane. I'm just going to write P for partially permeable, okay? Partially permeable membrane right there. These are water molecules. These tiny little blue dots are water molecules bouncing around. These green dots here are solute particles, dissolved solute. And we can see that if you look closely, let's zoom in a tad. Yeah, you can see that the water molecules are stuck around the solutes. That's because water molecules are polar. They have kind of slightly positive hydrogens and slightly negative oxygens, and they will orientate themselves and kind of stick to polar solute molecules. The hydrogens facing inwards if that molecule is slightly negatively charged, or the oxygens facing inwards if that molecule is slightly positively charged. So we kind of say that the water molecules are kind of trapped around the solutes. They're held. They're not free. So actually, if you look at here versus here, the left-hand side versus the right-hand side, there are more free water molecules, these ones, these little dots, these more free water molecules on this side than there are here. We call that really the water potential. So we have a high, and this is the symbol we give, high water potential here. And over here we have a low water potential here. So osmosis is the net movement of water by osmosis. Now that might mean that at any one point we might have a water molecule. Let's draw one here. I think we might have a water molecule that goes this way, but at any one point we're going to have more water molecules that are going this way. So the net movement of water is from high to low, but there are individual water molecules that are going either way. So that's osmosis, high water potential to low water potential. Now, in terms of units, the highest water potential is 0, okay? 0 is the highest. And that would be 0 measured in kilopascals, because it's a type of pressure, 0 kilopascals. That's pure water, okay? Pure H2O. So actually over here, it wouldn't be 0 because there's some solute molecules. And then low water potentials, very sort of salty or sugary solutions, we'd have negative water potentials. So something like, I don't know, minus 200 kPa, okay? That sort of thing, okay? Water potential is psi, you'd actually write psi equals minus 200 kPa. Okay, so that is that. Now, what about cells and osmosis? Well, we're going to look at two consequences when you put cells in different solutions. Okay, so this is cells in high water potential. Okay, and over here, down here, cells in low water potential. Okay, so cells in high water potential, we've got more water outside, more free water molecules outside, so water moves in by osmosis. We've got a plant cell on the left, animal cell on the right, so let's just write here, water moves into cell from high to low. Okay, so this is in an exam question, you'd write it a bit more fully. Water moves into the cell from an area of high water potential to an area of lower water potential through the cell membrane, okay, via osmosis, okay, via osmosis, got to use this word. So what happens? Okay, well, this cell over here, this plant cell, not a lot happens. It swells, it becomes turgid. That means swollen up, and that's because of the cell wall. Cell wall stops the cell from bursting. The animal cell, lysis, okay? It bursts. Can't stand being filled up with water. The pressure fills it up and it pops. So that's in high water potential. What if there's a lower water potential? So that means if it's like very salty or sugary outside. Well, this case, a bit more similar. Water exits. Okay, I'm not going to write the full sentence here. You might be able to write it. Something like water moves out of the cell from an area of relatively higher water potential to an area of lower water potential, out through the cell membrane. And what happens? Well, basically the cells become shriveled. Now there's two words for shriveled, different word for animal, different word for plant cells. So in the animal cells, we say that this cell is crenated, okay? And that basically means shriveled up. And in this one, we say plasmolysed in the plant cell. Now the significance of plasmolysed, why is it different from crenated? Well, it's that the cell wall maintains its shape, okay? It's tough, the cell wall. It just sits there, it's still there on the outside, maintaining its shape, but the cell membrane peels away from it inside. So let's just describe that a little bit more detail here. So cell membrane peels away from the cell wall. Okay, great. Almost there, last thing. For the last thing on this lovely sheet, we're going to look at a key membrane experiment, okay? A very important membrane experiment that you might do. Comes up in exams, so let's talk about it. Okay, so this is the Beetle Experiment. There's another experiment also that we probably could fit on this somewhere. I don't know where we would do it, but it could be about using osmosis to determine the water potential in a cell. Should we do that? All right, I'll very quickly sketch it. I've got a tiny little space here. It's going to be very minimal, but let's do it. Okay, so experiment. You basically have a graph like this, and you have different water potentials here, different solutions. Now you take, I don't know, a piece of potato, you core it, you try and get the all these cores of potato, let's say 5, 6 cores of potato, all exactly the same size, so same surface area, same mass, that sort of thing. And then you put them in different concentrations of water. You wait, same amount of time, let's say 20 minutes, and then you take the potato cores out. You blot them dry to remove any excess surface water, and then you weigh them, you record the mass, and you work out the percentage change. Here's the results. Okay, so this would be percentage change. And this is water potential here. Going from 0, let's say actually this is like minus the water potential. Okay, so this is 0, and this is getting more negative water potential this way. Actually, no, forget that. That's too confusing. Let's just say this is glucose concentration, okay? So with a low glucose concentration, your potato swells. It gains mass. With a slightly higher, it swells less. It swells less. And then you're going to get your results that are like this, okay? Those are the results, the dots. And then you join it up with a straight line. Da-da-da-da. Oops, that's not a very straight line. Let's get rid of that. Just about all right. And then it's this point here where it crosses that you can tell that is the water potential of potato cells. Okay, because those cells, if we look at these two diagrams, they haven't swelled, they haven't gained water, they haven't shrunk, they haven't lost water, they've just stayed the exact same, which means that whatever the glucose concentration outside the cell must be the same as the glucose concentration inside the cell. So actually, let's get rid of that. Glucose concentration of potato cells. Okay, that's one key experiment. Final key experiment. So this is the Beetle experiment. Okay, you take a piece of beetroot. Again, you do a core or disc. Piece of the beetroot. All right, maybe loads of discs. I don't know, maybe let's say 5 discs for each. Temperature. You got to wash these loads. Okay, you got to wash the discs like 3 times minimum in distilled water, and that removes the surface color. What is the colour, I hear you say? Well, it's this stuff, okay? In there's a pigment called betalain. And it's actually held within the cell, actually behind two membranes. We've got this tonoplast membrane, which is the membrane around the vacuole, and we've also got the cell membrane as well. Now, normally that pigment is held within the cell. So if you've taken these discs and you've washed them carefully, what you can do is then you can put those discs in some water, some distilled water, leave them in there for a period of time, let's say a minute, and then you take the discs out and then you look at the water. Has it got pink? At low temperatures, no. Okay, you measure that using a colorimeter. And you can see here that when we leave those discs in the water for a minute, let's say, at 10, 20, 30, 40 degrees Celsius, nothing really happens. It doesn't go, the solution doesn't go pink. But then suddenly above 50, 60 to 70, the membrane, well, the water starts to go really pink. Why on earth is that? And that's because around this temperature, the membrane structure starts to break down. And that, releasing the betalain. Okay, so if we go all the way back to our initial diagram up here, oops, when the membrane is hot, those phospholipids are jiggling about so fast that they're not really an effective barrier anymore. They're just kind of moving all about. They might pop out and they might disintegrate, or there might be holes between them where betalain molecules can temporarily kind of pop out through the structure. Okay, so that's everything on membranes. Let me just pull out here so we can get the whole picture. Yeah, so that's everything on membranes. Now that we've zoomed out so we can get the whole picture, we talked about the cell surface membrane, its structure, the fluid mosaic model. We've talked about the way things get across membranes. First of all, we've talked about diffusion and passive processes. Then we've talked about facilitated diffusion, still a passive process, but requiring a channel or a carrier protein. Then we've talked about active transport, sodium-potassium pump, and then forms of bulk active transport like endocytosis and exocytosis. Then we've gone back and looked at osmosis, which is a key example of another passive process, but specifically with water. We briefly touched on an experiment with potatoes and how you can look at osmosis using those, and then we looked at the betalain experiment, okay? I'm gonna link 2 videos underneath as well if you wanna go into more detail on those 2 key experiments, because we didn't really go in that much detail. So hope that's helpful, and can you please submit a picture of your lovely mind maps to me on Teams. Thanks very much. Bye-bye.